Now we're going to talk about how these thick and thin filaments are actually going to interact with each other, and how they couple, that filament sliding theory, right? How that actually occurs, and how the actual muscle contracts, right? So here we're going to get to this portion here, the thick and thin filaments. They're found in the myofibrils, and here is basically the cross section, right? And the functional unit, we'll see the sarcomere, right? And they basically blow up between these Z discs, right? We see the thick and thin filaments. The sarcoplasmic reticulum, as I said, stores calcium. The T-tubule, right, is what propagates that signal and what triggers the sarcoplasmic reticulum to release its calcium stores. And you see how much of it is around the muscle. So the moment the T-tubule gets the signal, it will trigger sarcoplasmic reticulum to release all the calcium, and you see how it's enveloped the entire, right, myofibril. So there'll be a lot of calcium release. So now let's talk about the actual contraction. Calcium is coming in here, right? Calcium is coming in here. So calcium plus is coming in here. Let's see how this actually causes the muscle to contract. So we're going to start at step number 1, right? So here we see actin, and that's the thin filament. So, the way I remember, I see TIN, actin. TIN, I think it's the thin filament. And even on the picture here, you see it's thin, right? As opposed to myosin, right? Myosin is going to be at the bottom here. Myosin is going to be the thick filament. And you see just the sizing of it, right? The myosin is also has this head, myosin head, they call it. Now, on the actin side, on the thin filament, we see these proteins. We see the tropomyosin, and we see troponin, right? So what are these? So tropomyosin, think of it as a chain, and this chain basically is blocking at this point right now. It blocks the myosin sites. You see how these red points where myosin wants to bind, it's not available because tropomyosin is like a chain that's blocking myosin from docking to it. When calcium comes in, it's going to bind onto troponin. So troponin is what actually binds calcium that's coming from the sarcoplasmic reticulum. The moment calcium binds to the troponin, it's going to signal the tropomyosin to move away from the myosin binding site. So calcium came to the troponin, signaling the tropomyosin to basically move that band out of the way. It tells it, I don't need you here anymore. You're blocking myosin head. Here we see calcium came in, bound, right, to the troponin signaling the tropomyosin to move out of the way. These now are exposed for myosin binding, right? Now we see before myosin can bind, we see this business of ATP, ADP, right? So ATP binds to the myosin head, myosin gets into the position, and through the hydrolysis, ATP is broken down into adenosine diphosphate. So this is diphosphate, 2 phosphates. This is triphosphate, adenosine triphosphate, and we see this inorganic phosphate when it's broken down. So ATP binds to the myosin head, puts it into the position. ATP is broken down into adenosine diphosphate and inorganic phosphate. So now, right, it's ready in, I would say, in the cocked position to bind and cause a power stroke, causing that actin and myosin filaments to essentially move, right? So here we see, right, this molecule goes away, and it supplies the energy. Myosin binds to the actin, to that site in specific, right? And it causes a power stroke. So power stroke, all it means is that it moves that actin filament inside, right? It moves it inside, causing those filaments, actin and myosin, to basically slide on each other, right? When this occurs, the sarcomere will contract, bringing both of those units, the contractile units, actin and myosin, together, right? So I'm going to explain this one more time, like taking each one of these pictures individually so that you guys understand. This is how we basically couple, right, the electrical transmission, the chemical ion transfer, to the actual coupling of the mechanical force of the muscle. So we'll go one by one, step by step, so that you guys understand, right? So here we see, right, step 1. Step one, you're at relaxed state, right? Muscle is, ATP comes in, it unbinds the myosin head to go into this active cocked position. So ATP came in, unbound myosin from actin site, and through hydrolysis, ATP is broken down into adenosine diphosphate and a phosphate, right? So it's in the radicocked position. Here we see the thin filament, which is actin, and here we see the thick filament, which is myosin. Here we see these chains of tropomyosin, which block myosin binding site, right? And we see this troponin here, right? So how this whole business begins, calcium comes in, right, from the sarcoplasmic reticulum, secondary to the T tubule, getting that electrical impulse. Calcium comes in and it binds to the troponin. The moment it binds to the troponin bound to calcium, it signals the tropomyosin to move out of the way. It says, I don't need you to no longer block myosin sites. So tropomyosin is moved away, exposing, right, the head. When this occurs, myosin head, right, which is here on the thick filament, will get rid of its ATP and phosphate and will bind at the site on actin, which is essentially the myosin binding site. Right? When this occurs, we see this power stroke. So, it will move the thick and thin filaments coming together, shortening that muscle fiber. Here, I just want to give you another picture, right? How all of this looks. So these are all these myofibrils. Here we see, right, the specific T tubules, right, over here, right, the T tubules, which propagate that signal. And specifically, we see that sarcoplasmic reticulum, which has all the calcium stores. So you see how much calcium is being released. So there's a lot of calcium that's coming in here to essentially provide that calcium for the troponin, right, to signal the tropomyosin to get out of the way. Right? So this is how it looks, right? Action potential came. The depolarization signal came to the T tubules. T tubules signal sarcoplasmic reticulum, calcium storage is released into the myofibril, right? Myofibril, each unit is here. So calcium is now rushing in, right? This is just another schematic. We see the T tubules again, we see the sarcoplasmic reticulum, and we see the myofibril here, right? So there's a lot of calcium that will be available for it to do its mechanical activity of contraction, right? And this is just how it does it. It has this calcium gated channels, and under the influence of the T tubules, they will open up, allowing calcium to come in. And once this is done, certainly there's calcium pumps that will move that calcium back into the sarcoplasmic reticulum to restore it, right, in order to facilitate that contraction one more time. So this is the signal that's propagating from the T tubule coming into the sarcoplasmic reticulum, releasing the calcium stores, right? This is actually a microscope picture, right? We see this is the tropomyosin, right? And this is the actin, the thin filament. This is without calcium, so it's blocking the docking site for the myosin. The moment calcium rushes in, you see how it opened up all these myosin docking sites. So myosin head can now easily, right, bind. So they say myosin can bind here, right? This combination of myosin binding to the actin is called cross bridging. So this creates cross bridges that I'll show you another picture, right? So this here, when it's bound, right, is what facilitates the cross bridging. The movement of the actual myosin head is what causes the sliding filament theory, right? The movement of that. But here we see the cross bridges, thin filaments, right? Thin being actin, thick being myosin. These here are the cross bridges. This is basically that myosin head that's now in a docking position on the thin filament, right? And you see how they look. That's why they call it cross bridge. And that's what creates this power stroke. This is essentially the movement of that myosin head causing the shortening, right? And this is again all the process together, right, from the beginning to the end. And then when you want to restart this process, ATP will come in. Where's the ATP made? In the mitochondria. That's why there's a lot of mitochondria in the muscle. ATP comes in, kicks myosin back into that cocked position, hydrolyzes into adenosine diphosphate, right? And essentially the troponin is awaiting calcium release by the sarcoplasmic reticulum under T-tubule control to restart this process. So this we just talked about initially, the process of impulse conduction, ion channel exchange, right, to the actual mechanical contraction of the muscle. Is there any questions at this point?
Hey, Nick, so I can see this happening quickly, like for a heartbeat, but how would a muscle stay in one position? Because this probably all takes place in a millisecond, right?
Correct, correct.
Stroke.
That is correct. So this takes place in a millisecond. So what happens is after that initial binding of acetylcholine esterase comes in and chops up that acetylcholine. So it's no longer on those receptors. So no more sodium can come in and depolarize, the end plate. So once it can no longer depolarize, right, it goes back to that resting potential, that negative potential, negative 70 millivolts, right? And the process restarts again. So you have a coupling of contraction and relaxation that occurs.
Okay.
All right. So let me see. I will show you another diagram of the exact same thing, and then later on I will do like a schematic drawing. Just I know there's a lot of moving parts here and a lot of minutia, so I'm gonna like distill it down even more simpler. So maybe then it will be more easier to understand. But this is just another overview of the same process. Maybe this will help you see it better. So this whole process called sliding filament model. Why it's called sliding filament is basically the two filaments of actin and myosin sliding on each other, right? Resting state. Resting state, no calcium is present. Why no calcium is present? Because action potential hasn't come, right? So this is in its form, right? It's not cross bridging, right? We see that the tropomyosin is blocking the actual docking points of myosin. So then the depolarization signal comes in, it depolarizes, it takes the resting state of negative 70 millivolts to, let's say, threshold, and then elevates it to the plus, right, millivolts. All this does is sends a signal to the T-tubules. T-tubules sense that signal, then they tell the sarcoplasmic reticulum to release the calcium. So now calcium is rushing in. As calcium is rushing in, the site of contact for the calcium is the troponin. The moment it binds to the troponin here, it signals the tropomyosin to essentially move away, right, from these docking stations, allowing the myosin head to bind to actin, causing a power stroke. This power stroke moves both the thin and thick filaments in together towards the M line, right, middle. And this creates the sliding filament theory. Muscle contraction occurs. Once the muscle contraction is done, ATP comes in, right, tells the myosin to go back into this position, disconnect from the actin, right, go back to the position. ATP hydrolysis, we see this adenosine diphosphate and phosphate now bound to myosin, awaiting, right, the next cycle, waiting for calcium to come in and start this process all over again. Okay? This is just a close-up view right here, right, of the same process that I was explaining, just a different graphic, right? Resting state, right? No calcium, and the moment calcium comes in, it causes this filament to slide, right? And this is basically, right, this is just a schematic, right? We talked about the sarcomere, right? The one functional unit of the muscle. Here we see the Z discs, right? And when this calcium binds, the myosin head comes to actin, and they basically pull this in together. So you basically have the contraction, the shortening of the muscle. This is actin sliding inside on the, right, on each other on the myosin. And this is done by these myosin heads that basically come and they cause this power stroke. Okay. So we see this combination of relaxed state, right? And then we have that contraction state. And what the tighten here is basically is these filaments that allow this to, when it relaxes, to go back into the position, like stretches it back for it to repeat, right? And this is essentially the sliding filament theory. Here we see the actual microscope pictures, right? We see the relaxed state, right? And this is the middle portion of it. And here we see when it's contracted, make it bigger, right? So relaxed state, we see the thick and thin filaments. And once it contracts, you see how they all brought towards the middle of each other. And this is done of the sliding theory model, right? Contracted sarcomere, and this is the relaxed sarcomere. The zone where you see that dark bands, what is the dark bands? Dark bands is essentially the overlap where the actin and myosin overlap. They're exactly on each other. The light zones, right, where you see this, the actin is apart, right? So you see, like, for example, right here, right? We see where the actin and myosin are on top of each other. This will be the dark zones. And the light zones is where it doesn't overlap, like the Z discs, right, and here. Right, and this is just, again, the whole picture together, right, from electrical impulse to ion exchange to the actual contraction. Right, and here, this, how it works, right, Ian, as you were saying, right, you have the stimulus, right, under somatic, right, somatic nervous system, which is part of the peripheral nervous system, under your voluntary control from the upper motor neuron, right, to the lower motor neuron. Then we have that phase of the contraction, right? You have the maximum tension, and then you have the relaxation phase, right, which is occurring after the release of the myosin head, right? Going back to the resting state potential, -70 millivolts, and then this propagation occurs again.